Okay. So should be able to see the banner.
Yep.
Great. All right, it's noon. Hello, everyone. Thank you so much for joining. Hopefully you're joining back from the last one where we talked about worms, fish, and flies. And today we have an amazing panel. I am super excited to be joined by these four awesome researchers. Talk about sort of a next step in a discovery to treatment lineage from these webinars that were generously sponsored by Biogen. So as I've said in the past one, and I'll mention again now, Biogen was going to sponsor our ALS Canada Research Forum. And due to the pandemic, we didn't have that forum. And then they generously said, well, let's come up with some ideas in terms of how we can continue education towards the research community. And so this was an opportunity to do something a little different from a typical webinar. And yeah, so if people have questions, you can type them in. We may not be able to get to them live today, but we have a lot to discuss. And but we'd always like to keep the discussion going because these things are, you know, all aimed at trying to ultimately get to some bigger answers. So without further ado, thank you to Biogen for this. And I'm going to stop showing my screen. You should hopefully be able to see our panelists. And so there's a bit of an echo, but I think we're going to work with that anyway. I'd like to introduce our esteemed panelists today. We're going to start with Dr. Tom Cunningham, who is a senior investigator at MRC Harwell, which is a Mouse Genetics Institute, Research Institute in the UK. He leads the mouse models of neurodegeneration team. Many of you will know Lizzie Fisher, who is involved intimately in developing great models of neurodegeneration. And now he's leading that team in what a great think tank over there, including you know, Adrian Isaacs, Pietro Fratta, work with Linda Greensmith, and really the forefront of developing new and better models of neurodegeneration, including ALS. So really excited to meet you, Tom, and having you here. Thank you. And just out of curiosity, what brought you to this? Was it just the fact that there was a lot of opportunity in neurodegenerative models to and new tools and new toys to have an impact, or was there something else?
My background is in developmental biology and genome engineering, and I'm relatively new to the field of ALS modeling, but I think what maybe hooked me in was a challenge that was given to me by Lizzie Fisher, who ultimately hired me 3 years ago, and it was to generate the new model of C9orf72 that we're working on, this huge repeat expansion, which is the most common form of ALS. So that's that as a genome engineering challenge, which really hooked me in. And so well, we can talk about that a little bit later, but it's something that we've eventually managed to figure out how to do, and hopefully we'll get those mice on the shelf in the near future. But that was, for me, that coming outside of a neurodegeneration background, that was the hook for me. But certainly, you know, it's obviously from a clinical standpoint, it's a big you know, it's an incurable disease, ALS, and it's something that just needs as many.
Great. Well, thank you. It's a little delayed, so I'm not sure that's my computer or
possible.
Okay, I think. So we also have a legend in ALS research and care, Dr. Terry Hyman Patterson, who is a professor of neurology at the Lewis Katz School of Medicine at Temple University. Also, one of the first multidisciplinary care physicians in ALS through, I believe, Hahnemann University and then Drexel as well. Recipient of a number of awards, including the Ford Norris, the Lou Gehrig Award, and the director of the Neuromuscular Disease MDA Center of Hope ALS Clinic. Thank you so much for being here, Terry. What an honor it is to have you. And also, I love your work. Is actually your work that I saw at a talk a while ago where you're talking about background of different mice and looking for genetic modifiers. It's like, oh, that's a cool thing to use ALS mice for to be able to look for. So I thought, what a perfect opportunity to have you here. So thank you.
Well, thank you. And I'm sharing the platform with my collaborator, Roger Scher, who also works in that area, and who is certainly an integral part of that work. And of course, my interest really grew from my clinical commitment to ALS and the fact that I am a clinician scientist. We are rapidly dying, of course. People who are able to both do lab work and do clinical work. I think I may be one of the last generations to be able to do that. But I became interested in, the heterogeneity of ALS, and then when the mouse model came out, I'm old enough to have been around when the G93A SOD1 first came out, and I was doing work, and we observed, depending on the background, there were differences, and we wanted to exploit the differences in severity by looking at different backgrounds, and worked, of course, with Roger and the folks at JAX. And it's been almost a lifelong commitment to find those modifiers and see if they translate to human disease.
Mm-hmm.
We'll talk about some of the problems because we use an SOD1 mouse, and of course everybody's going to wonder, will that really be generalizable to the sporadic or to the heterogeneous ALS population? I don't think anyone's sporadic, but I think that's a personal bias, that everything's genetic. And, it's all in our background, it's all in our genes.
Yeah, and I think the other piece I forgot to also mention was, as former co-chair of NEALS, you have a really great perspective on how these can translate to the clinic effectively, and then how, in some cases, they haven't translated effectively yet, and how some of the ways we can advance things to try to get there, to model the human condition better. Well, thank you very much. And as already introduced by Terry, Roger Scher at Stony Brook University, assistant professor at Stony Brook University, also previously worked at Jackson Labs as well. So very, very familiar with mouse models and mouse models of ALS. Now currently working on a lot of mechanisms of nucleocytoplasmic transport, retrotransposons, which we're not going to get into today. That could be a whole other thing, and genetic environmental connections. So Roger, thank you for being here. Was it actually the sort of the collaborations that brought you into the ALS field, or?
Actually, I was from an ecology background, and I happened to get a postdoctoral position at Jackson Lab with an ALS researcher, and the field of neurodegenerative disease just drew me in. And so that is where I got my introduction to it, and my interest in some of the genetics as well as genetic environmental influences comes from my ecology background thinking. And so I'm really interested in how can we use these genetic models to understand triggers as opposed to merely pure genetics, because the variability that we see in the heterogeneity of the disease in humans, as Terry mentioned, has to do with genetics, but it also has to do with exposomes, as the word is being used, and developmental biology, and when are we getting exposed to various kinds of triggers. And so that's where my interest came from.
Great, yeah, and obviously something that we're still really getting a grasp on in ALS, and how maybe even some of the learnings from the pathogenic mechanisms we learned from the genetic models could actually represent sporadic models. And I think that that's sort of one of the things you're looking at with the nucleocytoplasmic piece as well. It's very cool. Thank you. And last but certainly not least, the Chief Scientific Officer of the ALS Therapy Development Institute, Fernando Vieira. Really appreciative to have you here. I think from my standpoint, having someone from TDI here was great, because it really flips the mouse model on its head, in the sense of it's almost been like taking the idea of the lower ALS models, the worms and the fish, the high throughput, and saying we're going to do this in a mouse for the past 2 decades. and really some seminal publications in terms of guidelines and saying this is how what we've learned from extensive use of these, how you can try to avoid any kinds of biases in how your work is doing. Probably, and this is again, I should say these webinars, I'm totally, let's have open discussion. And if you have, I mean, I would imagine it's probably not been the most popular thing in times when TDIs publish things that have shown something didn't work or couldn't repeat. Let's say it couldn't repeat. But I think thinking from a patient organization standpoint, that's so important, because how many things have we had that have moved forward from mouse models or from smaller bits of evidence, and just could have been something that wasn't controlled for properly. An experiment becomes a multi-million-dollar clinical trial and ultimately fails. And that's not just about the money side, but also the side of the investment of the community in those trials and backing those. So I've always been fascinated since I was a PhD student from that model that TDI has. Excited that you have something that you've elevated to the clinical side now, because it must have been very rigorously covered. But thank you for being here, Fernando. I'll stop talking for a second. What was it that led you to TDI?
Well, happenstance brought me to ALS. I had an opportunity early in my career to be exposed to drug development, drug discovery for ALS. But it's a personal connection that has kept me in it. I ultimately became very, very close friends and roommates, in fact, with a person who we didn't know when I met him that he was a carrier of the mutation that also took his mother and his grandmother. But we learned earlier than we wanted to, the hard way. And he ultimately succumbed to ALS after a short battle. It was 9 months. And that ultimately inspired me to go full bore into this and dedicate my career to trying to identify treatments for ALS. And so that's kind of how I got into this, or why I'm still in it. With regards to the things that TDI has contributed, ALS TDI has contributed in terms of preclinical pharmacology and drug discovery for ALS, I like to say that we didn't come to solutions a priori, right? We just made a lot of mistakes very quickly. And we thought it was really important to share and learn our learnings with the entire field. So ultimately, the translatability of any of these models does remain an open question. I think each model needs to be taken in context. When Terry spoke to the heterogeneity of the disease and how it presents clinically, it's a really important point. In fact, I often make the argument that if you took a single person with ALS, a human being with the disease, and in some science fiction world you could clone that person would not be an adequate model of ALS, right? So every single model that we use in our labs, in our experiments, they all have their place. They all have their constraints and they all have their strengths. And we can execute on those models effectively. We control the things that we can control, and we try to put them in the context that we know to put them in. And I think that's really important. So I'm really glad that I get a chance to be a part of this panel today and have this conversation with folks who know a whole lot more about a whole lot of things than I do. So I'm looking forward to learning.
Well, if you feel like that's the case, don't worry. At least you're not the one on the bottom rung here for the 5 of us, because that'd be me. But yeah, no, thank you for being here. And actually, that's a great jumping off point, because I think, one of the things we would like to talk about at some point is, a lot of things are done in a single mouse model, and then it's considered almost good enough for some companies or whatever to try to elevate. And we don't have a second gold standard in a way in the field beyond what people sort of use as the G93A, but then, as Terry said, so representative of potentially such a small proportion. So I think we get into that maybe in a moment, unless people want to just bring it there. So as much as when we get to the future side, and I think that Tom's reviews actually on humanization are some of those things that are like, should be almost a required reading for a young scientist to say, look at the options that you can do, the things we can think about. I think when I think about, like, if I'm starting a mouse study and I had a graduate student, I would want them to read the paper, Fernando, that you were part of around, you know, okay, you've got power, you've got non-ALS related deaths, you've got litter effects, you've got sex effects with your male versus female mice, environmental effects, background if you're doing crosses, diet, aging. All of these things need to be considered to do a proper power, a proper ALS study. What did I miss? Because there's probably more. If you're, and then I guess the second question, and I would open this up to the floor, do you think most people consider all of those when they're doing a study these days? How about Fernando, if I started with you, since you're on that paper in 2008, and then also the guidelines, obviously, in 2010, the Ludoff et al. Paper.
Sure. So in the ALS space, I think that the field has gotten a lot better, right? So when I first came into the field in 2001, I think people didn't really fully understand the tools that they were working with, and didn't understand the types of variables that can contribute to spurious effects, both positive or negative. It's not always that you get a false positive result. You can get false negatives out of that too. I think, though, that a lot of groups with, you know, our publications from Terry, publications from Jackson Labs, Richard Mead's group at University of Sheffield. There are a lot of groups that are now doing pharmacology studies in G93A SOD1 mice at the appropriate rigor. And, you know, a lot of the sort of fee-for-service operations out there, like Jackson Labs, Charles River Labs, they're really doing a much better job. I think it is possible, but hard, to do a one-off out of an academic lab. I think that that's really challenging. I think you can line up the checklist of things to worry about. You know, balancing your study, distributing litter, figuring out protocols for copy number, but all of those things are hard and take a lot of work, right? So copy number assessment of the SOD1 G93A mice, it's not a trivial thing. And you can have a technician or a grad student or a postdoc with very skilled hands, and they know how to optimize those types of things, but it could take that person, you know, 5 months to really get that right. 5 months is a long time to wait before you think you are now ready to start getting the numbers of mice that you need to execute your study. So that's just logistically challenging in an environment that isn't optimized for it, like a place that can operate at a scale like JAX or ALS TDI or MRC, like those types of environments. But the good news is that those environments exist now. There are more of them than before. So there are various channels that a company or an investigator with an interesting idea or hypothesis could take their therapeutic and explore it with that type of rigor. And with that, I'll leave it for others to weigh in.
Well, you know, I can just adding to that, Fernando. You know, you make all the good points. I mean, you know, for instance, every male gets characterized in our lab, you know, for copy number, things like that. And so knowing how to handle it, but even aside from that, knowing what questions you really can ask with the model and what should we be asking of the SOD1? Why are we asking results of the SOD1 to generalize to a heterogeneous population? That, you know, So I think that one of the problems is that the mouse models and or animal models have all gotten a bad rep because they haven't always translated to human disease.
Mm-hmm.
But you're asking the model to do something that perhaps you shouldn't be asking it to do. I think Roger makes the point that these are good models for environmental exposure, and in fact, we have this very low copy number where there's only four copies of the SOD1 gene, and we had shown that copy number, the phenotype's dependent on copy number, and they don't always get sick, and so that's an ideal way to kind of look at environmental exposure and whether or not it can bring out disease, whether that genetic environment interaction results in disease. When I use the model, I have to ask myself, what question am I asking? And is this the right model to answer it? And I don't know that it's the right model to answer what's going to translate to a treatment in human disease. It may be, but I don't know, and I think it's wrong to malign the model totally just because it doesn't always translate. And I just like to make that point because, and for any postdocs or new PhD candidates, think about that. When you're designing your experiments, you got to choose the right model. Now, for oxidative stress, it's a great model.
Mm-hmm.
So
yeah, I think that's a really important point. That obviously, like the most obvious point is you can't test an ASO targeting C9orf72 in a SOD1 mouse, right? That's the most extreme model you can imagine. There are some things where you may imagine a therapeutic generalizes to some people, right? So there are hypotheses out there around oxidative stress driving disease, maybe in some but not all people with ALS, the immune system, et cetera, in different proportions as well, right? Like if you look in the G93A SOD1 mouse, you do a whole transcriptome analysis, you see disproportionate engagement of different pathways, and some are a tsunami, and others give you this small impact. Well, you may be able to target the tsunami and move the model, but you can't target the pathways that are minimally engaged because they're just a negligible contributor to the disease in that model. So you really do have to be mindful of all of this. I 100% agree.
Not only that, at different times in the disease, there's initiators and propagators. And,
yes.
And if you're treating an initiator and you're in the propagation phase, it's already triggered, horse is out of the barn, you're not going to catch the horse by locking the door to the barn. I mean, So
I think it also depends on which level of kind of the scale of the inquiry you're interested in using these different models. Like Fernando mentioned, places like TDI and Jackson and Charles River have the resources to do very, very large numbers that are actually looking at efficacy in a whole lifespan sort of way. A smaller lab, my lab, kind of an academic institution, I wouldn't even be considering that when I'm thinking of these models. I'd be looking at these models as a way to interrogate biochemical interactions or molecular alterations at a cellular level, and to use those tools for the appropriate scale at which I'm interested in asking questions that maybe could then lead to collaborations or outsourcing for those kinds of larger therapeutic testing. And so I think that the tools that we have are adequate based on the questions that you're asking of them. I think the problem has been historically is we've said we now have a mouse model of ALS, so now we can just use that as if it was a human population and find the answer that's going to cure it. And I think that that was naive, but understandable because we didn't really see 20 years of research that showed conflicting results or contradictory results. But now that we know all this, I think that the smart approach is getting better.
So actually, I would love to jump off the, oh, sorry, Tom, were you gonna say something?
I was just really going to say, you know, the other panelists have made all the great points there, especially what Fernando said. I think all models have their place, and it's about knowing your mouse, knowing the tools that are out there, and knowing your question, and really considering that before, you know, diving straight into the most convenient model that maybe the lab next door has or somebody has nearby. I think we even now have the tools to make the model for the question potentially. So yeah, there's a lot of consideration needs to go into the choice of model for sure.
Yeah, and actually this jumps off so many different ways. I would like to come back to that one about the model down the hall. But what Roger said also jumps off to me thinking about someone who wants to like write a grant application. These days, you know, there's always these drives by organizations like my own where you have this feeling of, well, you need to have some sort of translatability to everything. It needs to be focused on a thought process towards the clinic. Although, you know, if we don't understand the targets, that's a futile effort at some level. You need to understand the disease. So thinking about what Roger said, do you think that there is more, we're at a place now where maybe a lot of the individual lab work should be focused on the biochemical aspects of the disease, using the models to understand what that model can tell you as part of a larger piece of the puzzle. And when someone wants to look at, say, escalating a therapeutic target they've discovered into a therapeutic sense and trying to get proof of whether that works, that's when we should be thinking about farming those out to a place that does it better. Do you think that's sort of the niche we should get to in ALS, or do you think there's still room for someone to write the grant that says, look, I'm gonna test this particular drug in a mouse model and it's going to contribute to our global understanding, or is it just too difficult?
Personally, I wouldn't attempt to do go over the whole spectrum from basic biology through to the preclinical studies. I think collaboration is essential. You know, where I work, I bring the mouse genetics expertise to the equation, and we collaborate with Lizzie Fisher at the Institute of Neurology, and we have the neuroscience expertise. And I think really it's playing to different people's strengths is the way to go. Yeah, absolutely. If that's the question you were getting at.
Yeah, maybe I wasn't super clear in how I asked it, but you know, there are a lot of people who will say, okay, I have discovered this interesting thing in a cell line. Maybe I've done some primary neurons with the person that I know. Now I want to take that to the next level. Do you think that there is value in them in people trying to write something and say, look, I'm going to test this in a G93A, or should people, if they want to do that, should they be going through and trying to find who's the person that has a model that will best represent the pathogenic mechanism of what I'm focused on? Because I think there's been, as you know, a long time where somebody would just say, well, I know somebody who works in ALS and they do an SOD1 mouse model, I'll just do it in that, and then I'll get a publication out of it. But is that advancing us?
Please, Terry, you first.
Oh, well, yeah, we could both comment on this. You should choose your model appropriately. Just like when you come up with a potential agent, that's why we need biomarkers in people, because it's a heterogeneous disease. Everybody probably has a different mechanism that's driving disease. So you want to get responder populations. Mm-hmm. But I think in part, it's the life we live where if you do, this is where you can speak to it, Dave. If we write a grant and you're looking at a mechanism and you're saying you want to develop this as a pathway to modify, you're almost, you know, the first criticism that comes back at you if you're not doing animal work is, why aren't you doing it, you know, an animal model? And mm-hmm. Now choosing the right model is going to be appropriate for the mechanism, but we're almost pushed to that. And if you need the funding for your lab, looking for a collaborator might not be as attractive as attempting, even if you're not skilled in that area. And let me tell you, some of the grants I've reviewed, I'm sure, Fernando, you've had the same experience. people obviously have never used the G93A mouse model in the way they talk about it. Oh, I'm just going to do rotarod. Well, rotarod is really, yeah, you're laughing because you know. And Roger's laughing because we've all had rotarod experience. And, you know, but you can tell that they're just reading a paper and saying and parroting it. And they're almost forced to do that by the situation. You know, so I think in the ideal world, we would seek out collaboration and people would share funds and there'd be a more equitable distribution of monies so that everybody could survive. But there isn't, as a matter of fact. And so, I mean, I'm being very practical and direct about it as what I see the problem for that inhibits us doing things right. Playing to skill sets and playing, no question, that's the best way to do it.
Yeah, a couple of comments on the idea of what a grant application ideally might look like, right? And I don't know if these should be rules or hard and fast, but you know, yes, I would look to see if the person, number one, was thinking about the appropriate model. Now, to be fair, you know, regulators really like the SOD1 mouse model because regulators are a little bit lazy about the way they're thinking about it. They tend to lag a tiny bit behind the science. So if you are thinking about trying to move a drug into clinical development, you know, the FDA is interested in seeing a SOD1 mouse. They might be interested in seeing a TDP mouse too, or profilin, but they are interested in the SOD1 because it has often been used. So that is something of a constraint. And I think that's fine. I think the SOD1 mouse has a place, but not for everything. But I think it does have a place. As far as grants go, yeah, it would be great if people would acknowledge that they found a partner, right? So that operates at the scale, a collaborator of some kind. And if they don't, then you know, I would be looking to see whether I wish I could ask these questions. We don't really have this cycle of back and forth as a reviewer, but I wish I could ask, do you plan to breed, you know, piecemeal and cobble these, you know, 42 mice together over a year and a half? Or are you going to do an in vitro fertilization and generate a batch of mice where you can do appropriate litter matching and run a study, you know, concurrently? The latter I would view really favorably, right? Because they would have been thinking about it critically, and the study would have the appropriate, you know, all of the controls and the treatments would be happening at the same time and under the same conditions. So it's that type of thoughtfulness that I would seek, but unfortunately we don't really get that opportunity to look for that or even suggest that in grant reviews.
And honestly, most academic situations, you don't have that technological capability to be able to do all that unless there's a mouse core at your facility that allows you to do that. So I agree with what Terry and Fernando were saying, and from my perspective, I wouldn't be putting in a grant where I'm proposing that I'm going to do everything in-house. If I was doing it in a large scale testing of a drug that I had an interest in, because the scale and the cost to have that many mice, to house that many mice, and to have people to be able to do all of the experiments in the time that it would take when you have to grow the mice up and you have to age them and all that, would probably, from an institutional cost perspective, be more than getting a grant that says that I have this and I'm collaborating with an institution that can run it at a cost of scale that is going to be half of what it would be if I did it in-house. If I was at Jackson Lab, I'd probably have a different perspective. But at an institution that doesn't have that sort of mass facility, I wouldn't even think about that, but I would, if I was working on something like a potential compound, I would be writing grants that said that the money is going to be, you know, divided, and I'm going to be using this as part of a consortium grant where I'm going to be hiring out one of these institutions to run the trial, and I provide the agent of interest. And in the meantime, I would do in-house cell culture and mouse model assessments of cell biology and biochemistry and things like that to parallel the holistic work that would be going on in the full rodent model itself.
So actually, so let me just, okay, so this panel has said, and I'm going to paraphrase this, but I know it's not true, but I'm still going to say it anyway. We are now at a time in ALS where the study in a small n, slightly 0.0497 p-value, slightly increases by 1 week at G93A, get you a publication, but it's not impact. This isn't where we should be going as a field if we want to start to think about translating the understanding we've got and getting that towards a therapeutic or clinical development realm. Those things are probably not the future of that. That's we're going with that. We're going with that. So I have a question though, or if I'm wrong, just say no. But so back to the G93A and flipping that now to, let's say, a lot of the work that's being done would relate to RNA biology now, or to, you know, DPRs versus my gosh, why am I blanking? What's in the nucleus? My gosh, so the C9.
RNA foci.
The RNA foci, my gosh. Told you I'm the lowest on the rung here. But okay, so now let's take an example of somebody who has something and they want to look in a TDP43 model, or they want to look in a C9 model. Now it becomes even more complex. What one do I choose? Well, I know somebody who has a TDP43 model, but they're so different, and we have not got beyond the SOD1, sort of that gold standard idea of something that, you know, they all kind of provide a different aspect, and possibly some of them maybe even should be retired. Although I said that, maybe no one else wants to say that. What do you do? What do you do if you want to? Is this still a situation where it's like, if you want to look at a therapeutic sense, you should be considering collaborating with somebody who knows the models well enough to know which model is best for testing your particular avenue of therapeutic? Or do you think that that can be done? Like, do you think there are a lot of studies where people are doing it and they're just picking a TDP43 model because they know the person who has it and they trust that person? And no one wants to say it, right? But
I'm sure that's happening. I'm sure that's happening. I'm sure that's happening, that people are picking a model because they know someone, and you know that might be their only option as a route to you know, someone who's starting out. They may not have a large network, but they happen to know somebody. That's what they would do. But you know, it's not an easy answer. Which mouse do I pick? It takes a lot of consideration, and like Roger said earlier, are you at the preclinical end, or are you at the basic biology end? You know, in the TDP model, if you're interested, are you studying you know, phase separation, or are you studying cytoplasmic mislocalization? Different models have different pathological aspects, so you need to understand what those aspects are and whether it fits with your hypothesis, I guess.
Yeah, and I mean, I can't remember which conference it was last year, but somebody was talking that when they say that 95% of ALS patients have TDP43 proteinopathy, mislocalized aggregates, one of the pathologists said, well, yes, but they're very sporadically found throughout the brain. You don't see it everywhere. You only see a few cells that actually have that. You don't see those in non-ALS patients, but you do see it. But then there's mouse models that don't have any TDP43 mislocalization, and yet they still get progressive paralysis and death and many other splicing defect alterations, even though the TDP43 never mislocalizes out of the nucleus. We have some of the new CRISPR-mediated knock-in point mutations, where they've actually just made the mouse gene allele have the polymorphism that is found in the human patients, and none of these seem to be getting sick. They have maybe some cognitive defects, but they don't get ALS. And so this tells us something very different about rodent biology and lifespan versus human biology and lifespan, and what is the cumulative burden that leads to neurodegeneration in human aging. And so I think that these are actually opportunities for us to intelligently use these models to ask the questions that we can now ask. Whereas before, without this variety, we were asking questions of models that weren't appropriate. But now that there are many different models out there that have different aspects and different features, I think that actually allows us to, instead of just taking the one from down the hall, which unfortunately occasionally will still happen, most people that I talk to don't do that. They're using the model and using what they find in the model or what other people have reported in the model as a, they're not just using it as a hammer. There are, 30 different kinds of hammers, and each of them are good for a different kind of job. And I think that people are starting to recognize that the toolkit is diverse, and therefore the questions that we can ask are becoming stronger.
There's even another level of complexity to that, because you can take those same animals and breed them on some of the newer backgrounds that have been developed at JAX, the collaborative crosses.
Yep.
And you see a difference if you breed it, that same mutation on a black 6 or an SJL, which are divergent, but then you put it on these the collaborative crossed animals or outbred crossed animals, and there's a huge disparity, much more like the human population in terms of heterogeneity. And that should be considered, because that probably would be a better model of human disease, even from that mutation. We all know that all of these mutations, C9 patients don't always manifest. SOD1 patients, you could have siblings with SOD1, and one sibling will develop the disease and one won't. So, so there's another level of complexity yet to determining the best model and how to use that model.
So I agree that it's good, because people are being forced to be thoughtful about how to use the models. And I do think lessons learned around SOD1, for folks who have been around, we do try to apply those lessons learned when thinking about the next mouse, right? Like, and the much maligned SOD1 mouse, actually, it turned out it was a pretty good model in terms of replicating motor neuron degeneration and paralysis. And none of these other mice, or very few, have really been able to do that in these other transgenics that have emerged. But people can use them different ways, and that's heartening. The downside to it is hard. It's harder to get independent replication of people's work, and that might play out a couple of different ways. Eventually, people will get, if you start to get interesting hits that start to translate a little bit, people will flock to those pathways and those models, which will be good. The other possible good thing that could come is multiple models generating the same answers, right? Mm-hmm. So multiple models giving indications of efficacy by targeting the same pathways. But I think we're in early stages with this, and I don't think that there's a shortcut to saying, okay, let's go all in on this one and standardize around it. I don't think we're there.
Okay. So, Tom, I wasn't sure.
Oh yeah, I was just going to add, it's also about managing expectations for your mouse. Roger was talking about the transgenic models versus some of the knock-in models that were knocking the point mutations directly into the mouse gene. And with those more physiological models, the mouse is living 2 years. You shouldn't necessarily expect those mice to get a full-blown ALS. And those models really are more for getting to perhaps the early mechanisms, early cell biology, some of the early changes, rather than studying the disease process itself, the full disease process itself.
And I think that's also a good point, that everything we know about the pathology is from post-mortem in human patients. And we don't know what the process that leads up to that during a lifetime actually is. And so biomarkers are obviously one important aspect, because that can potentially tell us about something that's going on in the central nervous system. But I think that one of the benefits of the animal models is to study how these genetic, because all of these obviously are genetic models. We don't have a sporadic model. But how do these genetics that we know lead to human ALS in another mammal, in the mice, how can we use that to investigate developmental biology and aging biology in the central nervous system that has an insult that in humans leads to ALS? And I think that the models are good for that also, is not to necessarily model an endpoint of ALS, but model a cellular replication of progress. And so that's another way of thinking about these models, is that we're not necessarily looking at them as a true stand-in for human, but rather as a platform for investigating the things we can't investigate in humans. Because we wouldn't treat people with a drug 30 years before they develop the disease, unless we had a great biomarker that tells us at 20 that you're going to 95% chance you're going to get it when you're 50. But we treat the mice when they're 2 weeks old and show that the disease is better. Well, that's great, but that's not necessarily the right approach.
Right, unless it's like ASO for pre-symptomatic.
Right.
Eventually that'll be the case, but yeah, it's much more difficult in the wider population. Okay, so taking this in though, and rewinding this and thinking about, there are a number of, we've learned a lot in ALS in the last 5-10 years in terms of pathogenesis, things that we wouldn't have grasped maybe in the previous 2 decades, probably because we didn't have as many puzzle pieces to connect, right? Let's say someone has that, now putting on the funder hat, thinking about the various societies around the world that maybe want to have impact with their dollars, but also want to make, like, you want to make sure you're not just constantly trying to drive things on the clinical end when we don't have a great understanding of the disease still. It's still very complex. How could the field, from a larger sense, try to help drive good therapeutic targets, interesting potential therapeutics into proof of efficacy through using rodent models or whatever, like coming out of cells and say a fish or a worm or something? How can you think of something outside of the box or something that could be done to try to help push that, so that we don't continue with this same thing of just one lab testing one thing? Is it just nurturing the concept you've all said about trying to collaborate with the right people to do bigger studies, more like a human trial where maybe you've got a mouse that's outbred, or the collaborative cross, large enough number, double-blind, placebo-controlled in a mouse as opposed to a human? Do we drive those things, or do we just have to let things go as they are and kind of keep funding little bits here and there, and we'll get there? Like, what is there something that could be done that's a sea change in how we approach these things?
Well, you could do a platform trial in mice. I mean, you could go and you could say, here's a basic mechanism that is operant in people based on a particular biomarker, say oxidative stress, because we know SOD1 drives oxidative stress. And you could set up the platform trial in a large lab like TDI or JAX of various pharmacologic agents that are directed at oxidative stress, looking at which one, depending on how you want to measure effect, be it any or all of the above, progression of disease. You started at onset, which is pretty much standard what we recommend anyway. And Roger, you mentioned people started early. Right now, most people do start it at onset. Starting drug at onset, looking at survival, looking at some whatever behavioral assay you like, and pathology, and run it, like the platform trial in humans is being run, directed at a single mechanism and proof of concept of that mechanism. So, kind of looking at oxidative stress, for instance. Maybe you'd look at inflammation. If you're doing an anti-inflammatory, and pick the model which has the most inflammation, which may not be the SOD1. There are models that actually have affect inflammatory cascades more. So that may be a novel way. You could look at several drugs and take the one that has the best effect based on whatever criteria you determine that makes sense, of course, and then try to get a responder population in people to match and go for it. And I don't know. It kind of might be
It's hard, right? So looking at it's a little bit easier when you're looking at drugs that are already approved for other indications, right? You have opportunities to kind of leverage known information about dosing levels that are effective, and whether or not the drug gets into the CNS and all of that. When you're talking about new molecules, then it gets much more unwieldy, right? Because then you have to do the pharmacokinetics experiments, and then you end up having to sort of optimize dosing around pharmacodynamic endpoints biochemically, or at a molecular level or at a cell biology level, and at a tolerability toxicity level, and then you triangulate, and by the time you're done with all of that, now you can execute your efficacy study, whatever that might look like. So if we think that treatments for ALS, that meaningful treatments for ALS can come out of the existing sort of FDA-approved pharmacopoeia, then that idea has, I think there's potential there. If we think that it's mostly going to come from new chemical entities, then that becomes a bigger challenge. And those are open questions, and it's worth having conversations about that.
Well, that just goes to creating a model that will work for your hypothesis, and thinking about the models that you use.
Yeah.
But
exactly.
There's a lot of repackaging of drugs going on now. And
there is. It's true.
Well, see, this is the question I had. Is that, so thinking about, like, there's for years, obviously, at the person living with ALS level, there's always the drive of, faster, whatever's in trial, we got to push it faster. But from a basic research standpoint, most of what's in trial is still generic to some extent. And this because it's being repurposed for 7 different things. Now, something like AT-1501 came from ALS, right? It's the ASOs are directed at ALS targets. So we have a lot of things that are being learned at the basic research level that are more ALS things, come from a C9 model, a FUS model, and they get to that point where they still need to be proof of efficacy in whatever model, rodent, whatever's going to be most accepted, to get there to the edge of the valley of death. So not even at the valley of death where we're talking about all the work you have to do to get it to phase 1. It's just, is it already on track, or is there something we can do to help speed that process up? Because I'm also mindful that people need their publications and they want to do 5 different things before it gets to that stage. But from a patient standpoint, when is it going to be that we get the smart ideas that are directly coming from a C9 model, for example, past that stage? Like, I don't know. I mean, maybe I think that's sort of what Fernando was saying, is that once you start to talk about the new things, it gets much more complex, because it's what models, what is it telling us, how are we going to get there? And I guess thinking from a standpoint of a patient organization, is there anything we can do to accelerate that?
Well,
Or are
so
we? Yeah.
I have an example, right, that I'm struggling with, and I'll throw it out there. In fact, Tom, when you mentioned the C9 mouse, I was like, I got to write to you right after this, because so we discovered that there's a possibility that methylation of dipeptide repeats, particularly arginine residues of dipeptide repeats, might play a key role in the toxicity of those dipeptide repeats. I don't know if the dipeptide repeats are relevant. We really don't. But in the model we built, it was, and inhibiting asymmetric methylation of those arginines mattered, right? I don't have a mouse model in which to test that. I can look at Len Petruccelli's lab, and they're using AAVs to overexpress PR, I believe. Might be GR, might be both. And I think, oh, well, maybe I should call up Len and see if testing a PRMT1 inhibitor in that model would show efficacy. But maybe Tom has a different model that I could test in. Like, I'm at this exact nexus with something. This obviously isn't, if it works in SOD1 mice, it would be weird, right? Like, it's not the right model to test it in. But I need that model. So it's on me, right, as the researcher, to find the solution to that. That's a responsibility. I have that responsibility to patients who live with repeat expansions, to get that answer in, to do it right. So, and it's not to do it wrong just to check a box so that I can make it happen faster. I need to do it right faster. So suggestions are welcomed.
Okay. So, yeah, so it's a difficult question. Is basically, and I guess if maybe one thing I'm gathering from this is, if there's anything that those in our organizations can do, it's maybe facilitating more and more connections between researchers, so that reaching out from Fernando to Tom, for example, is just easier, because it's not a person you've never met. Or, I mean, I guess not for both of you, probably it wouldn't be a big deal, but I'm thinking about maybe a postdoc who says, well, I really like the model that Clotilde has, or Laura Random's C9 model or something. I really like that one for what I'm doing, for specifically. So, but I don't know her, I don't know them, so my PI doesn't know them, I can't reach out to them. Maybe it's providing more opportunities for the global community to just see that people are receptive to these things. I guess if you have money, they're very receptive to these things, to collaborate on a project, but that it's difficult, and we probably just need to facilitate more connections, I guess. So.
One of the things would be in the review. I mean, you see all the grants, and looking at people who are working in the same space and connecting them together and trying to develop consortium that work together, and sort of funding the consortium to enable or to fuel that collaborative effort. Because that's always going to be the underpinning. You'll have people who have good funding and people who have not so good funding, and so sort of building those consortiums and helping everybody, shoring up the foundations and having them work on the problem together and take working to their strengths, as Tom would say. So that everybody brings different tools to the table and having them work together. I mean, that may be a way, and not sort of picking one as the most likely horse to win the race, but rather making a team based on the interest that's expressed by the grants that you're reviewing and put them in barrels. I'm just throwing that out there. It's like spaghetti scene, what sticks to the ceiling.
Right. And from a global perspective. Sorry, Roger. Go ahead.
No. Just a thought that I had previously that is kind of related to this is if you're, what Fernando was talking about, new compounds and things like that. I have friends here at Stony Brook who are in chemistry or in physics, and sometimes we'll get together and we'll be talking and describing the different kinds of work that we do. And sometimes what they say resonates with something that I've read about in ALS, but often some of the things that I talk about from the biological standpoint, they had never heard of before, they had never thought of before, but they are like, well, it's weird. My grad student is actually working on so-and-so. And so I think within institutions, if there's ways to certain places already have this, like TDI, which is all about ALS, but to get the biologists together with chemical engineers and biomaterials specialists and things like that in conversations about here's the state of the field, and here's what we haven't been able to succeed at, and are there other fields that we as biologists really have or computer scientists who came in to do electrophysiology that have no idea about what the others are thinking or what the others have knowledge of, and finding ways to build within institutions, maybe an ALS science group as opposed to an ALS biology group, because we tend to think in terms of making connections with other biologists. But if you look at the Venn diagram, we probably have a lot of overlap and not as much that makes us unique and separate when we're all biologists. And so I'm wondering, is there a way for us to bring in other fields that can make changes that we would never think of?
So globally, we need to unite people more. We need to get people more collaborative, but in a way that provides an opportunity for it, because everyone's very busy and things that we can maybe do globally. Okay, so I would like to, because I realize it's already been an hour, and I thank you all for doing this. Want to get on to the future of stuff, future of mouse models, the future of models. I do want to ask one last question that's bothered me for a while, which is, do you think that all mouse models that would eventually be used, for therapeutics or efficacy, should show some effect by riluzole or other things that get more time-tested proof? Let's say a Darvone gets, like, if it does get real-world long-term proof, or other things that could come along that could eventually be approved, like now with recent evidence that the real-world data of riluzole, that maybe it's a little more effective than we've been giving it credit for. Do you think if a model has absolutely no effect in that, it should be used as a therapeutic model for humans? Or is that okay? That's my question.
So, please.
No, please, Fernando, you go ahead.
No, I was going to ask for clarification on the question. So you're saying that if a drug, so I'll start with this. Riluzole in our hands, if you combine 3 well-powered survival efficacy studies together, does give us a 4% life extension
Okay.
in the SOD1 mouse. You have to, that's statistically significant. You have to really beef it up to notice, but it did happen. And now, on the other hand, edaravone, radicava, does not. At 2 different dose levels that we tested, we couldn't eke anything out of it. Does that mean that neither of those, does that mean that, so one would say then, okay, the mouse model was predictive with riluzole, not predictive with edaravone. I think that there is room for drugs that don't work in SOD1 mice to work clinically. I do think there is. You need to have other evidence, though, to justify then its testing. It can't be, you know, an underpowered SOD1 mouse study that gets you to clinical testing. You have to have other reasons to have advanced it.
I think what you're asking, David, is that, you know, should a mouse model, to be a good model for therapy, have to demonstrate first an effect utilizing the drugs we know work in the human population?
A positive control,
Is that
yeah.
what you're really asking? And
Well, so I'm not asking it just of the G93, though. I'm asking it of a theoretical platform. Sorry, go ahead.
No, Dave. I didn't mean to troll the G93. Should any mouse model that we develop to be a valid? I think that's what you were asking.
Except for the fact that, I mean, there's the caveat that we haven't really parsed out whether or not riluzole has an effect in people with SOD1, I don't think, from human studies, right? So it doesn't mean that that study isn't valid for people with SOD1 mutations and who are humans. I'm thinking about, yeah, I mean, it's a good point. Do we have any positive controls, or we just simply don't? And if we get them that work more robustly, will we need to incorporate those into rodent model studies to represent whether or not what we're seeing is likely to work in humans?
I don't think you would need to, but I think it would be nice to.
Okay.
Yeah.
And I think that you would, unless the milieu changes, I think the FDA is going to insist on some animal work. They seem to do that now. So again, it goes back to choosing the right model, you know, to support what you're doing. And, you know, people are the best model of the disease. And
right.
we can't just go right to people. And they're not even a good model, are they? There's huge variability. I mean,
well, right. I mean, I think this
problem until we could segregate populations into bins, because it's not one disease. Go ahead, Roger. I didn't mean.
No, that's exactly the point that I was going to make with the heterogeneity. These animal models are models of genetic ALS, and they may have unique biochemical pathways that are reflected in the humans that have those genetic mutations, but there may also be common pathways among various genetic ALS and sporadic ALS that riluzole may have efficacy in. And so if you do riluzole in a huge human population of ALS patients, maybe it's more effective with certain genetic forms, but it may also be that the sporadic people who responded well may have underlying genetic modifiers that we aren't aware of, or it may be that they had particular kinds of, you know, exposures growing up that caused a certain pathway to become insulted, maybe mitochondria versus something else. And so riluzole is more efficacious against that. And so I think the heterogeneity question comes in as to, do we need to have these be perfect models, or can we use them to try to find genetic specific versus sporadic generalized impacts of some of these drugs, and use the mouse models to kind of tease those things apart?
So it goes right back to knowing what questions you can ask of the model. And look, in, you know, what you didn't mention is that in edaravone, And of course, my clinical hat comes on. In Adarago, what they did was develop a responder population based on their initial trial or their initial phase 3. It didn't reach statistical significance, much like our mouse experiments. And they went back and they said, let's look at the people who, quote, responded. What did they look like? What was their phenotype? Because they certainly had no biochemical markers or others, because they didn't combine it with measures of, oxidative stress, or 3-nitrotyrosine, which I think would have been a better marker. But anyway, they went back and they said, what's the responder population? Then they do the trial with the responders, and they were able to tease out an effect. It wasn't washed out with the variability in putting everybody together.
But other drugs do that too. For drugs for heart disease and high blood pressure, they found that there are specific genetic polymorphisms where it works in 9% of the population, but the big trials that started out didn't show any impact. But the population of ALS patients is incredibly small compared to the population of people with high blood pressure. And so the numbers that we have available for these kinds of things to tease apart those responders versus non-responders is a more difficult situation.
But it's not even, there's not even, because many of these trials are driven by pharma, and there's not a lot of money to do the studies in conjunction that would help them tease that out. The biomarkers of, target engagement and effect of if something's supposed to reduce inflammation. I mean, neurons trying to do some of the markers in the CSF of, inflammatory cascades and cytokines to go along with it. And they may, when they do their second order analysis, say, oh, look, the people who had the highest level of X were the best responders. That's not integrated into all trials. And until we start doing that and looking at responder populations, and then going back to the animal models and saying, the animal model that's going to predict in this responder population is this model. I mean, it's sort of a two-way street.
Yeah, coming back.
Human disease can inform what we do in the animal model, and the animal model can inform, but only if we collect the data that will enable us to do the two-way. So.
Okay, so mindful that we're down to like the last 18 minutes, I want to talk about the future of mouse models and or other models. So Tom, I'm going to you because I loved your review articles on humanization, and I learned an awful lot from there, both scary and exciting, because there's things in there that I'm like, okay, I think about, there's the AD models where when you put the human cortical neurons in there, they get susceptible, but the mice aren't. And I wonder, well, how good is the mouse model, etc. Okay, so first of all, are mice the future? Or with the tools and the toys that we have now, will it be rats? Will it be pigs? Will it be, do you think it's still mice because we're so engrossed in that?
I mean, I think for some of the complex modeling, I think at the moment really we're more limited to mice. So we can do CRISPR in other larger animals, but if we're some of the large humanizations, for example, we humanize C9, it's 25-30 kb, and then we've got to go through the embryonic stem cell route, and so there isn't really that route with other animal models. But I mean, certainly that would be beneficial to be able to move into a larger animal model, if that technology came on board. But I don't really think we have that at the moment. But I think in certain parts of the world, CRISPRing is going on in whatever pigs or sheep or other animals, and I think that there's benefit to that for sure. But then it's obviously the resources required are huge compared to mice. So I think mice is still going to be the staple for genetic modeling, mammalian genetic modeling.
Do you think that we are going to continually be evolving the models that we have now that there's so many tools, AAV, CRISPR, everything that we can do, the different backgrounds? And if that's the case, do we have to relearn what we've learned in these more advanced models? Or do you think it'll just become a validation, a very short validation of what we already know to make sure that it's sort of on par? Do you think there's a chance that we're going to learn completely new things because we've got more advanced models?
Well, we don't know the answer to that yet. Certainly a large part of it is we're making our humanized mice. Do we see the same things that we see in the transgenic? I don't know the answer to that. And it's possible that we'll learn new things, but I don't know the answer to that.
It's all speculation. I know. Okay, so then what about someone so let's say right now we have mouse models or animal models of, I don't know, like 10 different genetic causes of ALS.
Mm-hmm.
Is there still valid reason for people to be doing more of the ones? Like, for example, someone comes out with a NEC1 model, or you have a, there's TPK1 models now, but I'm thinking about CCNF models and all this. Are they going to be used in the field to the same extent, or do you think most of what we're doing is going to be stuck with TDP43, FUS, C9, SOD1, and revolving around those four? Like, if someone were to make those models, will they have uptake for use, or again, is it just the choice of the pathogenic mechanism someone's looking at?
I mean, I think the more rare mutations aren't going to have as much uptake. There's not going to be as much interest. There's still benefit in making them. It would provide a better representation of the spectrum of potentially completely different diseases, different mechanisms. But for sure, the uptake for the more rare variants is going to be less. It's to be expected.
I mean, I could make one comment on this. So a relatively rare variant is profilin 1, right? Mutations in profilin 1. But there have been a couple of transgenic profilin 1 mouse models that have been developed that have shown relatively robust phenotypes that actually look a lot like the SOD1 phenotype. And a difference, though, is that they do show TDP pathology, TDP43 pathology. So a lab like ours at ALS TDI, where we do have a scale where we could, optimize a model that has similar characteristics to the SOD1, and imagine sort of the pharmacokinetics and pharmacodynamic work that we do in the SOD1 G93A sort of being done across platforms on another model and running in parallel. That's almost scalable for us. So looking for a mouse that might be a rare genetic variant, but is different enough from SOD1 and might capture a different part of the Venn diagram that is ALS pathology and pathophysiology. Running those in parallel, we think, would be useful. So we're trying hard at that right now. It's a heavy lift. Both just work and resource-wise to get there, but we're trying to do that lift. If another one were to emerge, we would probably try to do that too, just because of the way we're built and the team we have and what we're good at.
It's almost like having a platform trial where it's not different drugs in the different arms, but different models in the different arms.
Exactly.
Which then would highlight which mechanism might. That's actually a great use of these other mutations and models, because the problem is the variability in disease duration in some of these models. So you have some that live 250 days, but if you make them homozygous, like the dynactin mouse, which nobody talks about, but dynactin, if you make it homozygous, you almost have it in a reasonable time frame. Heterozygous, they live forever, and they develop disease. But just tweaking each model so that they can be somewhat comparable in terms of how long you have to run the study, and then you might actually normalize it by % increase in survival.
Yeah, something like that. Exactly.
Normalize it on something, but
and from a
must be a novel platform, and then you say, oh, this drug works best on people with these mechanisms that are operant, and then you go look for that responder population in people and translate that.
Yes.
Yeah, and I mean, in parallel to that, I think you could also look at it in terms of, instead of having different models from different genetic forms, different C9 models or different TDP43 models show very different pathology. And so if you ran 3 different TDP-43 models in parallel with the same drug to look at not just the, I don't want to say merely, but not just the physiological changes like, you know, rotor rod and lifespan and things like that, but cellular biochemical, immunohistochemical changes. That might tell you something about how these models have similarities and how these models have differences, which could tell you more about TDP-43 and how it is involved. And so I could see where you could do both of those kinds of things. They're asking different large questions, but I think that they can both give you some very important answers to why do we see such variable phenotype with different models of the same gene, even sometimes the same point mutation.
Yeah.
I mean, you'd expect that if you have SOD1 G93A, it would be different than G85R. But why would 2 different labs that both make a TDP-43 A315T have such different, even if you put them on the same background, right? And so even accounting for copy number. So these are the kinds of questions I think that these kinds of platforms could also, you know, address.
Yeah, I mean, the C9 knockout's a great example too of something that just different places, completely different situation. Okay, so that's, I mean, that's a lot to take in. I think there could be a lot more discussion moving forward. I'm also mindful that we're down to the last 9 minutes and it goes so fast. So I would actually like to do something different. I want to go around the panel and I want to say, what are you most excited about with respect? So, okay, I'll preface this, sorry. Models of ALS get a lot of flack. There's a lot of people who say we don't have good models of the disease, and that is why we also default a lot to the G93, as Fernando said, because it's reliable for a motor neuron degeneration, and you know that's what we're ultimately kind of aiming for. There's a lot we can learn from it, and it's straightforward in a certain way. Not but there's a lot of flack that comes about not having the right models. But as individuals who care about this and work on this, do you feel that's true? And what are you most excited about with the future of modeling ALS with animals, with rodents, with whatever? So I'll go in the reverse order of alphabetical. So Fernando, just a minute or two each.
Sure. So I am most excited about the conversation that we all had and the consensus around the idea that each model has their place. There is no one perfect model, and that's not just limited to rodents. That's everything from, you know, induced pluripotent stem cells, worms, flies, fish, mice, and up to humans, right? There's no one model that will adequately address the complexity of ALS. There is an acknowledgement in the field that that's true, and there's a desire in the field to use the right models for the right questions. So that's exciting to me.
Great. Roger?
Well, I agree with what Fernando said. I'd say that what's exciting to me is the conversation really within the past 5 years, but accelerating, is that ALS is not one disease, and the treatment for ALS is probably not going to be a simple cure where you just give a single pill and it not only stops the disease, but you regenerate all of your nerves. It's going to be treating it like a chronic disease, slowing the progress, allowing quality of life, allowing extension of lifespan. And that I think that the animal models and the iPSC models and the worms and the flies, now that we are starting to come to that viewpoint in the field, is going to allow us to start to find multi-drug targeting opportunities that we maybe were not necessarily thinking about before.
Great, great personalized medicine idea there as well, and this could be a whole discussion unto itself with the various big data, PMP, all of these things going on that we could start to piece together, and biomarkers obviously to understand who's going to be responsive to what. But that, you're right. Maybe the breadth of the mouse models and the right mouse model is actually a good representation of the fact that not every human is going to be the same, which is basically what you've all been saying this whole time. So, Terry.
Well, it's really the same kind of thing. I mean, ALS is not one disease. Why would you expect one model to mirror it? And getting that across to the critics of the model so that people start to understand what the models can and can't do, what each model is good for. And now the additional excitement, if you will, is having tools that we can create even new models that are more specific for the questions that we want to ask. Site-directed mutagenesis, CRISPR, the outbred models that Jax has developed for really then creating a genetic heterogeneity, which more mimics the human condition, and more education for the public and for researchers on how to use these models in a productive way that maybe will lead to good science and answer and address some of the questions that will get us to good, drugs, and biomarkers, understanding mechanism and how to target responder populations, and how to actually develop markers that will help you bin people into responders. So it's really reiterating what Fernando, Roger said, and I'm sure Tom will have to come up with something new. Go ahead, Tom.
No one ever likes to be the last one. But I concur with everyone.
You see my list, it's all those things. But I mean, yeah, the personalized medicine aspect I think is very exciting. And speaking personally, someone who makes tools, just hopefully being able to provide those tools that people are looking for to really get a good representation of what's needed for the spectrum of disorders. And we do have the tools to answer the question at hand, like Terry just said, and I think that's exciting to be able to really make these bespoke models rather than say, oh, what's the best thing that's out there, and maybe if it's not the best thing that we really need, but it's what you've got. But now really there's maybe no excuse other than funding to say, well, if you need a specific model, you can almost make it. If we can manage to pull off making a C9 expansion, we could probably make anything. And it may be scary to think about humanization, but you could even imagine combining humanizations, whether it's Staffman2 and TDP43 and those kind of examples, then that's quite an exciting prospect.
And I didn't mean scary as in any negative way. I mean, just it's an exciting read because there's just the tools have exploded, and so what we're learning from it is just massive in a short time frame compared to decades. It's just getting faster and faster. So I think that's sort of what I meant. Also because, as I said, I'm not as informed as all of you, so for me, maybe a little scarier than all of you, where it's exciting. But thank you all. Really, I really appreciate this. This is exactly the type of thing I was hoping to get out of this, and I feel like more of these types of discussions are really helpful. If I'm thinking back to when I was a student or a postdoc, it would have been awesome to be able to hear these things, because these are the type of perspectives, the type of conversations I'm not going to hear. I'm only going to see a talk, or I'm gonna try to glean what I can from the literature, but I may not get that kind of wisdom that you've all been able to help provide here. I'm sure you had hours and hours more wisdom, but I'm sure you're glad you don't have to share them with me today. But I really do appreciate your time. It's very valuable. Thank you to Biogen for allowing this opportunity to kind of have these types of discussions, and to the audience out there who have been listening. We hope that some of this will have been valuable and can help us get to a better day where we don't have ALS as an untreatable, or let's have it as a more treatable disease in the foreseeable future. So thank you to all of you very much. And for anyone out there that wants to attend the third one of these, we'll be talking about iPS cells, and I have a lot of work to do. Get myself ready for that panel. But thank you all again, and wishing you all a wonderful rest of the day. And Tom, wonderful evening, and hope to see you all again in person someday soon.
Yeah.
Thank you.
Thanks, David.
Take care. Stay safe, everyone. Bye, everyone.
Bye.
Thanks. Bye.